So I'll introduce our next speaker, who's Julia Carnevale. She is one of our also newest faculty members, I think the absolute newest, and she's an assistant professor at UCSF. She received her MD at Harvard Medical School and completed her residency and fellowship in Hem-Onc at UCSF, subspecializing in GI oncology. She's worked with Alan Ashworth and Alex's lab to use innovative CRISPR screening approaches to identify novel therapeutic targets for GI malignancies. She's been awarded a Damon Runyon Physician-Scientist Training Award, an AACR-ASCO Young Investigator Award, and a Burroughs Wellcome Foundation Award. And her lab that is just starting up will focus on using knowledge of key target genes to reprogram the biology of T-cell therapies against aggressive malignancies. So take it away, Julia.
Hi, everyone. I'm excited to be here. And today I'm going to talk about my work essentially to harness the discovery power of large-scale CRISPR screens in primary human T cells to identify genes that we can manipulate in those cells to alter their biology for therapeutic efficacy, and then testing those strategies in preclinical models of T-cell therapies to ultimately nominate those strategies to move towards clinical trials in patients. So when we started this work, it wasn't feasible technically to perform large-scale CRISPR screens in primary human T cells. So after trying a number of different strategies to circumvent this challenge, we ultimately landed on transducing the human T cells with the sgRNAs, with high efficiency, and then introducing the Cas9 protein with nucleofection. And ultimately, we found that this strategy led to high knockout efficiencies using single guides, and ultimately that this was readily scalable to genome-wide libraries. And once we were able to edit genome-wide libraries in human T cells, we then moved towards performing large-scale screens in these cells. And the first number of screens we performed were essentially taking those genome-wide edited pools of T cells and then re-stimulating them and basically labeling them with a non-toxic proliferative dye, CFSE, and then using that to sort based on their CFSE profiles for non-dividing or highly dividing cells. And ultimately, after next generation sequencing of these different pools of sorted cells, we were able to generate basically lists of genes that were presumed to either enhance or abrogate the proliferative capacity in human T cells. And after we performed a number of different validation experiments on those lists of top genes that influence proliferation in T cells, and we showed that majority of those genes did, in fact, boost or inhibit proliferation as expected, as well as boost T cell activation. But perhaps the most exciting validation experiments we performed, or further experiments we performed, were actually taking human T cells, transducing them with a TCR specific to NY-ESO, then editing them for single guides against top genes of interest from those screens. And then co-culturing them in an arrayed fashion with tumor cells that have been labeled with RFP, and then using a live cell imaging system over time to essentially monitor in vitro what the tumor cell clearance capacity of those T cells were over various time points. And so this is just raw data off the machine, but essentially what we found was, in fact, a number of our top target genes, when edited in these T cells, were actually leading to improved tumor clearance in vitro over time. So this is exciting, suggesting that we could take the output from these screens and use this to really inform how to manipulate T cells for higher anti-tumor efficacy, which had clear relevance for immunotherapy T cell therapies. So after we published that data, we actually performed a number of different forms of these screens with the same pipeline, but in essence, adding modeling various immunosuppressive factors that would be kind of found or modeled in the tumor microenvironment. And basically doing a macroanalysis across a number of these different screens, cell-based, cytokine-based, different inhibitory molecules. When we looked across these screens, there were really only a handful of genes that were converged upon across these screens that had 5 or 6 that all 5 or 6 different screens agreed upon, where these genes, when knocked out, led to relative resistance to suppression of proliferation in the presence of these suppressive factors. And so what one of the genes that was particularly interesting was a RAS gap, RASA2, that's never before been studied in the T cell or immune cell context. And what I'm showing here in panel D is RASA2 is a member of the GAP1M RAS gap family. And what you can see here is when we look at the guide performance across the GAP1M family members, RASA2 really does stand out as being significantly enriched and across donors and across different guides in these screens. And I show here a RAS GEF, RAS GRP1, that's well described in terms of its role in proximal signaling for T cell activation after T cells are stimulated. And that's well described. But interestingly, the particular RAS gap that's functioning to curb RAS signaling after TCR activation is not well described. And you can see here that this data suggests that it may be potentially be RASA2 that's most kind of important in curbing RAS signaling after T cell activation. So we went on to perform a number of different array-based validation experiments to show that indeed, when we knock out RASA2 and then subject these cells compared to control guide edited cells to these different soluble suppressive factors that we originally tested, we do see that there is a relative resistance to suppression of proliferation, shown here in pink is RASA2, as well as to tumor cell clearance. And the most complex screen that we did was this cell-cell co-culture screen with Tregs. And I'm showing some data here where essentially when we do a three-way co-culture, so we take the effector T cells and edit them for either control guide versus RASA2, and then co-culture them with RFP labeled tumor cells, and then add in different proportions of matched Tregs. What you can see here is in the control setting, the control edited effector T cells control the tumor cells fairly well. But when you add in increasing ratios of Tregs, these effector T cells start to lose their capacity to control the tumor cell growth in vitro. However, this does not seem to be the case or is much more subtle for the RASA2 knockout T cells, where they're able to really maintain very impressive tumor control despite the presence of the Tregs. So all this is just to show that we used kind of convergence across our suppression screens to nominate a small list of genes of interest. And then we use various validation studies to show that indeed it does appear that in the setting of RASA2 knockout, these T cells are relatively resistant or boosted in the setting of these suppressive factors. So this nominated RASA2 is a particularly interesting gene to study in the setting of T cell therapies. So then we dove deeper into understanding the biology of what's happening when we ablate RASA2 in these T cells. And so what essentially we found overall is that RASA2 seems to be a negative regulator of RAS signaling after TCR activation. So it's TCR stimulation dependent in terms of its negative regulatory function. So what you can see here is that when we give the T cells activation, the RASA2 knockout T cells have more active RAS activity. And there's also, this is mirrored in various assays showing increased levels of mediators downstream to RAS. So phospho-ERK here showing a time course. Overall, the kinetics of activation are relatively similar, but there's a higher kind of peak activation of phospho-ERK, also higher levels of phospho-MEK and phospho-S6. So would be expected downstream of RAS to be more active after stimulation. Higher level, these cells also are able to secrete higher levels of cytokines, multiple cytokines, effector cytokines. And this is data just to kind of highlight that this advantage seems to be very stimulation dependent, both in terms of signaling, activation, and proliferation. Really, the advantage is only seen in the context of stimulation. So we dove deeper into this kind of effect of enhancing sensitivity to antigen. So what we think is happening is when we delete RASA2 in these cells, we're effectively increasing the gain on these T cells, making them more sensitive to any given level of antigen. So what I'm showing here are two kind of parallel experiments where we do a titration of either CD3, CD28 bead exposure, or in this case, NY-isopeptide. And what you can see here is that for any given antigen level, there's a relatively higher phospho-ERK response to that antigen. suggesting again that these cells are more sensitive to antigen. And we also looked at this in the CAR T cell context. Here we took CD19 CAR T cells and we co-cultured them with NALM6 leukemia cells with varying levels of CD19 antigen density. And using annexin as a readout, what we can see is that in the very low CD19 NALM6 co-culture context, there's the kind of highest delta in terms of the annexin levels in response to this co-culture, suggesting again that these RASA2 knockout CAR T cells are more sensitive to low levels of antigen. So we also went on to look at the effects on transcriptional programs in these RASA2 knockout cells. And first we used a set of Jurkat reporter lines. And we found that when we knock out RASA2 in these cells, we see higher levels of these transcriptional programs that are really relevant to T cell activation, AP1 and NF-kappa B. We also did RNA-seq, and what was fairly unexpected in this context was what we saw that was most kind of different between the RASA2 and the control guide T cells was many genes involved in mitochondrial biogenesis and OXPHOS, and we thought that this might just be kind of an expression profile that's seen in highly active T cells, but we also looked at the CBL-B knockout T cells, which are highly active, and we didn't see this kind of pretty impressive signature of a number of key mitochondrial biology genes. And so this was interesting and not expected. And we went on to do some functional studies and found that using MitoTracker and Seahorse, there was indeed higher levels of mitochondrial mass, both in CAR T cell and TCR T cell models. And there's higher levels of oxygen consumption in these RASA2 knockout cells, both basal and maximal. So both at a transcriptional and functional level, it does seem that the kind of metabolic set point of these cells is different, which may relate to their overall fitness. And I'll go on to show some of their kind of fitness advantages in various assays in the next few slides. One more note on this slide, I'll say that we're also interested in looking at the endogenous levels of RASA2 in T cells. And I'm not showing it here, but our data showed that when we activate T cells acutely, the RASA2 levels actually go down. And then when you expose them chronically to repeated tumor stimulations, the levels of RASA2 rise over time to actually a higher level than their baseline. And so we looked at external data sets and found that this was actually mirrored in external data sets as well. So this is a mouse model of acute and chronic or acute infection, a bacterial infection, and also liver tumorigenesis. And we found that after acute infection in this data set, RASA2 levels appear to go down. And then in this data set, we saw that in the acute versus chronic phase, there's higher levels of RASA2 in the chronic phase in this tumorigenesis model. We also looked in a large data set of human blood donors where T cells were isolated and stimulated, and this is about 100 human donors, and it looks like, once again, the acute stimulation in both CD4 and CD8 T cells leads to downregulation of RASA2. We also looked in a data set across 2 different solid tumor models, looking at the TILs, which is the peripheral T cells, and found that indeed the TILs had higher levels of RASA2. So this is all kind of suggesting, when we put it together, that RASA2 may kind of be naturally downregulated by T cells after acute exposure to antigen to kind of boost their own response. But then over time with chronic stimulations, the level actually rises and may act as a sort of internal checkpoint to kind of shut down some of the potent T cell activation responses. This is a hypothesis, but it is interesting that the endogenous levels of RASA2 does seem to be regulated based on activation, both acutely and chronically. So we saw very clearly that these RASA2 knockout T cells are very active, very potent killers. They're very much in an effector state, but we wondered whether knocking out RASA2 would actually make these cells more vulnerable to becoming dysfunctional, more exhausted over time or after repeated stimulations. So to look at this, we essentially developed this repeated tumor stimulation assay that other groups have, other speakers today have referred to, where we take human T cells, we transduce them with either TCR specificity or CAR, and then we we basically edit them for genes of interest, and then we expose them to tumor cells with the matched antigen on the matched MHC, or the CAR-relevant antigen, every 48 hours over and over again with fresh tumor cells at a 1-to-1 E/T ratio. And these cells can be isolated after any given number of repeated tumor stimulations and characterized both phenotypically and functionally for what's happening with those cells. And so this is just some data to look at the assay itself, not specifically focusing on RASA2. What we can see is the viability of the T cells tends to go down after repeated stimulations. The various exhaustion metrics, CD39 here, tox tends to go up. Cytotoxic molecules tend to go down. Oxidative phosphorylation profiles go down. And potentially, most importantly, this right upper panel, the capacity to kill tumor cells, so the functional readout of these cells does seem to go down quite clearly after repeated stimulations. So you can see after 1 tumor exposure, there's really good tumor control. After 3, you start to see loss of that tumor control capacity. And then after 5 to 6 stimulations, these T cells are really dysfunctional in their capacity to control tumor cells. So this assay is kind of a nice readout, and I see that multiple speakers are using this readout. So next, we did this with control edited versus RASA2 edited antigen specific T cells in this system. We didn't see overall any clear difference in the canonical surface markers for exhaustion. So at the very least, these RASA2 knockout cells are not more exhausted than the control cells. We saw higher levels of phosphoric, not surprising, higher levels of activation responses, and higher levels of multiple cytokine production across the different T cell models. And also higher levels of just a panel of cytotoxic relevant molecules. I mentioned this already. I just put this to also mention that after repeated stimulation, we do see this higher level of the oxygen consumption rate and mitochondrial mass as well. So the functional readout was the most striking, I'll say. So after the first stimulation, there was a slight advantage in this donor to having RASA2 knocked out in terms of tumor control. But after 6 repeated stimulations, these control T cells really kind of become dysfunctional. But the RASA2 edited T cells are just very, very persistent in their capacity to kill tumor cells. And this was true over 7 different donors, collapsed here by area under the curve analysis at a number of different E/T ratios. The delta between the control and the RASA2 cells is very consistent. And this was also true in the CD19 CAR T cell model as well. So overall, while these are very effector, very kind of potent T cells, they're also very kind of persistent in their functionality. They're resistant to becoming dysfunctional in this chronic stimulation assay. And so next, we wanted to see whether this translated to in vivo models of tumor control and survival. And so we use a number of different models, both TCR T cell and CAR T cell models in liquid and solid tumors. And so on this first row here is the NY-ESO TCR T cell model using both A375 melanoma model here in B, and then a NALM6 leukemia model that's been engineered to express NY-ESO here in D. And what you can see is in both cases, there's increased tumor burden control, and there was also increased survival, particularly for the A375 model here. This is a very, very aggressive model. It's a little bit harder to see the survival advantage here. And the most striking model was one that we performed in partnership with Justin Aquam's lab. And what you can see here is that using his CD19 CAR knock into the track locus model with and without RASA2 knockout, is there's a very, very striking advantage in terms of tumor burden control in these mice, which translates quite clearly to a survival advantage as well. And we also partnered with the Crenshaw lab at St. Jude that has a number of different solid tumor CAR models. And in their one of their models for sarcoma using FA2 CAR, we also saw an advantage in terms of tumor burden control and survival as well. So this kind of effort across models showed that indeed knocking out RASA2 in the TCR CAR T cell context, liquid and solid tumors does seem to have this clear advantage, and it's for this reason, a very exciting novel target that may be very, very relevant to translational efforts in the future. This is very new data I got yesterday, but returning to that CD19 knock-in CAR model, we wanted to look at the bone marrow of these mice. So we collected the bone marrow at day 16 and looked at the level of NALM6 tumor cells and also CAR T cells, and found that in a satisfying way, you can see lower tumor levels, as you might expect, as well as higher levels of CAR T cells kind of persisting over time in the marrow, which is nice to see that these T cells are persisting at higher levels. And we also did a competition assay where we basically engineered the cells to have an EGFR-T in either condition, and then mix them at 50-50 ratios with either the control of the RASA2 without the EGFR-T to look at the competition in the marrow. And you can see quite nicely in either of the combinations that the RASA2 knockout CD19 CAR T cells does outcompete the control T cells here at day 16. So all to show that there is this kind of persistence phenotype so far in our data as well. And this is my last data slide, but this is looking at the kind of overall safety of this strategy. And in the studies that we perform, we've seen that overall, the RASA2 knockout T cells are still cytokine dependent. Here's a withdrawal data showing that the T cells lose viability if you take away IL-2. They're also dependent on antigen in terms of their killing capacity, using here an A375 wild type co-culture system, and their capacity to secrete multiple cytokines. It's all antigen dependent. And then we basically took 2 cohorts of mice and gave either just CAR T cells alone or CAR T cells at clearing doses, and then monitored those mice over time. And we saw that these mice had similar body weight, similar CBCs, and on histopathologic analysis of the marrow and the spleens, we didn't see any evidence in any of the cohorts of any differences in terms of lymphopoiesis. And just generally, this data shows that overall, we don't see any kind of clear understream proliferation or evidence of stark transformation in the studies we've done thus far. So in summary, the data I've just shown you suggests that, first of all, I'll just point out that what's nice about these 2 stories is that we can take this kind of comprehensive unbiased screening approach in human T cells, and then that can kind of point us towards novel targets that you can then drill down a little bit more deeply to start to understand how regulating these targets in this T cell therapeutic context can really change the biology of these T cells. And in this case, it looks like it has clear therapeutic advantage. So it's kind of an exciting example of how the pipeline can point to novel and therapeutically relevant biology. So in summary, analysis of multiple CRISPR suppressive screens really converged on this RAS gap, RASA2, that had never been studied in immune cells before, and nominated it as a loss of function target to confer relative resistance to various suppressive factors. And then we found that RASA2 ablation in T cells boosts signaling downstream of RAS and increases their effector function. Overall, this kind of increases the sensitivity to low antigen levels. And increases canonical transcription factors of T cell activation. And we also found, surprisingly, that knocking out RASA2 does seem to change the metabolic state of these cells. And given the kind of increasing literature around the fact that oxidative phosphorylation, mitochondrial function may be quite important in the long-term fitness of T cells, particularly those in the tumor setting. This is interesting and potentially relevant. We found that endogenous RASA2 levels are actually acutely downregulated upon T cell activation, but then seem to increase after chronic stimulation, suggesting that by knocking it out, we may actually be kind of co-opting a natural mechanism. But by keeping RASA2 low, we're kind of taking out a checkpoint to ongoing functionality of these T cells seeing repeated antigen stimulation. Showed that RASA2 ablation appears to confer resistance to dysfunction in the setting of this kind of chronic stimulation model. And then most importantly, RAS2 ablation in these differing preclinical models, both TCR-T, CAR T cells for both liquid and solid tumors, does seem to confer an advantage to these cells in terms of tumor burden control as well as survival. And so I'll just take a minute to recognize and thank my excellent mentors and all the wonderful members of the Ashworth, Marston, and Atrium labs that have helped so much on all of this work, as well as Giedrich Krenschut, who has worked with us on on that sarcoma CAR model, and as well as all my KO8 advisors and collaborators, and all of my incredibly important funding sources that have funded so much of this work. And I'll stop there and take questions.